Welcome students. So, we were discussing the applications of UV visible spectroscopy in this course biomolecular spectroscopy. So, if you remember what we discussed in the last class, how the structure dictates the function, that means the intrinsic structure of a chromophore that determines what is the absorption frequency or the wavelength of a chromophore. Like if you have a more conjugation, then longer wavelength, that is called bathochrome shift. So, depending upon how much conjugation it has, what kind of delocalization of the unpaired electron have, many of such factors, unsaturation basically dictates where this chromophore is going to absorb. That is called, like shifting towards longer wavelength is called bathochromic shift. Then we also looked at the environmental factors play a key role in the absorption frequency, like in which solvent the experiment is recorded, what is the pH of that solvent, what is temperature of these solvents. All of these basically significantly dictates what is the energy gap between the ground state and excited state, and because of that your absorption frequency of or wavelength will change. Other than that, we have seen that, like we have saw an example how pH can act as a chemical shift for any ionizable oxochromophore. The solvent polarity can also induce the red shift or blue shift depending upon what kind of transition we are seeing, whether pi to pi star or n to pi star, because what it happens that because of this solvent polarity, the delta E can little bit change, either decreases or increases, the gap increases or decreases, that essentially changes the absorption frequency of the UV visible spectrum. Then oxochrome, we have seen oxochrome enhances the absorption. So, any attached group, right, which increases the conjugation. Conjugation increase, that means absorption intensity or even the wavelength increase or decrease, this is called hyperchromic shift. So, last we concluded with many terminology that we have seen, what is bathochromic shift, what is a hypsochromic shift, hyperchromic or hypochromic shift, and how these are essential for analysis of a chromophore, and chromophore essentially is a reporter of the phenomena that is happening. So, using these shift in the resonance in the absorption frequency, we can learn a lot about the environment of the chromophore. Now, we also looked at under these factors allows us to analyze the experimental conditions, predict absorption pattern, interpret the UV visible spectrum in the instrument that we use in the laboratory. And because of that, we have developed many applications centered around a simple technique of UV visible spectroscopy. So, we will extend our discussion in this direction and we will just look at the multi-component analysis. Why? Because you see, in any substance or a biological molecule, it could be possible that more than one chromophore are there. So, signal is coming from one or multiple component. If it is coming from multiple component, then the same spectra is a sum of two or more reporter chromophore. Now, in those case, you have to, if you want to understand the property of a particular chromophore, we have to deconvolute it. So, in those case, this curve fitting program that are there, they come into play and they basically helps us in differentiating the contribution of one chromophore versus another chromophore, right. So, that is the modern curve fitting technique helps us in developing these analytical tool. So, essentially as we know, absorption is proportional to the number of molecules that absorbs the radiation at a specific frequency, and it is even true if more than one component are present in in the solution. So, what we do? Let's look at this concept of something called principle of additivity. So, if there are multiple component, how we can do the quantitative analysis based on this principle of absorption at any wavelength that is contributed by more than one chromophore. So, what it says, principle of absorption at any wavelength of a mixture is equal to the sum of absorption of each component in the mixture at that wavelength. So, suppose two or more chromophore are contributing towards absorption at this particular wavelength, right, or at this particular wavelength. So, here the total absorption will be given by the absorption of component 1 versus component 2, and similarly at other wavelength we have the absorption contribution coming from component 1 and component 2. So, let us see how we can write it. So, absorption of a mixture at each component will be sum of absorption of each component at that wavelength, which in turn depends upon the extinction coefficient of that chromophore and concentration of each component. So, here it is a simple algebraic equation that you can see. So, this says the absorption happening at lambda 1 will be given by a extinction coefficient of that component A at lambda 1 multiplied with concentration of A plus extinction coefficient of B at that lambda multiplied with concentration of B. And then at another wavelength it is the same, some multiplier B, extinction coefficient of A at lambda 2 multiplied with concentration of A, and then extinction coefficient of B at lambda 2 multiplied with concentration of B. What that mean, if you just go back and explain it again. So, at this wavelength, say here suppose we are saying that black is sum of the two absorption. So, at this wavelength lambda 1, how much blue and how much red is contributing will be resulting as a this black curve, and similarly at this wavelength how much blue is giving, how much red is giving, that results into black spectrum. So, at each wavelength the contribution coming from chromophore 1 and chromophore 2, the concentration as well as molar extinction coefficient, that we can add it and come up with this equation that I was discussing, right. So, now we have a two equation, two unknown, and we can solve it. So, say equation can be solved something like that. For determining the concentration of each component that is contributing. So, C A is the concentration of component A, that will be A lambda 1 minus A lambda 2 multiplied with extinction coefficient of B lambda 1 divided by extinction coefficient of B lambda 2, and this is the denominator, this is the case. And then concentration of B can be again solved like a similar like what we did for A. So, essentially it is solving any algebraic equation where we have two equation, two unknown, and we can find it out if we have experimentally determined the absorption, we know the molar extinction coefficient and like a molar extinction coefficient, that is all you need for getting the concentration of A and concentration of B, right. So, in a simplistic term that is what we can solve it and get the contributing concentration of each of these component. Let us see now here in a pictorial form, I am seeing these two spectrum, say yellow and green. So, yellow is something like this, and yellow where it comes and absorbs maximally at this lambda say 230, and green is like there which absorbs at lambda 210. So, resultant spectra because of these is red one. So, in this case green is contributing maximally or maximally at 210, yellow is contributing maximally at 230, right, that is what it is. Now, in this case there was no major overlap, but what happens if there is a major overlap? That means both of these yellow and green are giving some absorption at the both wavelength. So, here you can see Yellow is giving higher at 210, but something as well at 230, and similarly for green here and here. Now, in this case, because of significant spectral overlap, the two component analysis will look like this. So, red will look like this. So, at 210, the quite a bit contribution coming from yellow as well as green. At 230 also quite a bit contribution coming from green and yellow, right. So, in this case we have little bit of difficulty in analyzing. Here it was very clean, because at this wavelength 210 wavelength maximally it was coming from green, at 230 maximally it was coming from yellow, but here not the case, both are contributing. Because of this contribution, a little bit of deconvolution is required to understand the contribution, how much coming from green, how much coming from the yellow. Great. So, now suppose if there is a mixture of many components, say let us take a hemoglobin derivatives. This is these are all hemoglobin derivatives that contributes to the spectrum between say 450 to 650. So, here if you look at these are the different component, oxyhemoglobin, here 2 absorption, that is a yellow, one around say 490, another around 570, right, something like that. Then you have a sulfur hemoglobin which is in red, carboxyhemoglobin again 2 peaks, the hemoglobin which is in orange it's minimally absorbing, and deoxyhemoglobin this is there. So, these many if you take these all components, and if you are taking a blood where most of these components are, many of these components are there, the analysis of such case becomes little bit cumbersome, and you require a multiple statistical tool for this multiple component analysis, like PLS, partial least square analysis, then principal component PC or PCR regression, or multiple least square component MLS. So, for such cases where many components contribute and more than one wavelength contribute, this analysis becomes complicated, and therefore, lots of analytical tool needs to be developed and tested how you can get the contribution of this. You understand where we are. So, if we take a blood, in blood many of these things can be there, like oxyhemoglobin, deoxyhemoglobin, the normal hemoglobin, carboxy, so all these are possible, and in those case analysis becomes cumbersome. So, here lots of data analytics tool or data science comes into picture in biomolecular spectroscopy. Great. Now, we have looked at the solvent, effect of solvent on the absorption spectrum, right. So, one example just I want to show how and why solvents affects the value of lambda max, shape of the spectrum. So, you look at this molecule spectra of biologically important compound, phenol measured in isooctane and ethanol. In ethanol you look at, this is the broad spectrum, however, in isooctane you see a clear 3 peaks. So, many times essentially the solvent plays an important role. Now, in the case that we were discussing, the local effect of the solvent is significant in getting sharper or the narrower spectra, and therefore, experimental design also becomes quite crucial, that what is the solvent to be chosen, what should be polarity of those solvent, so and so forth. So, now we looked at the basic principle of biomolecular spectroscopy, UV visible spectroscopy. Let us look at what are the important biological compound that have absorption. So, you see NAD or NADP, these are like nucleotide bases, absorbs at 260, this is the molar extension coefficient. NADPH and NADH, they are at 260 and 340 molar extension coefficient. Then FMN, you see FMN structure, lots of conjugation, or FAD, they are 260, 375 and 445, 450. FMN is like a extension coefficient and is higher than the FAD at both the wavelength, and then pyridoxal again 250 and 320. So, these are the compound, biological active compound that may bind to protein and give rise to fluorescence at a different color. Similarly, cholesterol is a nice compound, right. Cholesterol is found in the various membrane composition. This is important membrane composition for membrane fluidity and rigidity. This again has a UV visible signature at 235, calciferol, beta carotene, retinol found in eye again absorbs at 330, then octadecanoic acid, all the bases ATGC, all of them around 260 and they have different molar extension coefficient. See, these two guys has two rings, right. So, their molar extension coefficient is higher than those which has one ring. Now, other than these which is present in the biomolecule, there are prosthetic groups. These actually they are tightly bind to the protein. It can be a small organic molecule or metal ion known as prosthetic group, and essentially for biological activity something like heme is a prosthetic group. So, heme is in oxidation state +2 when it is bound to oxygen, right, and that is what we have. The oxyhemoglobin and deoxyhemoglobin, you can see solid line for deoxy at 550, oxyhemoglobin one this line is splitted, right. So, splitted one around 570 and another around 540. Now, this kind of ion in blood you can have the oxyhemoglobin as well as deoxyhemoglobin. In those cases, if you want to determine how much oxygen saturation, like a pulse oximeter we had looked at, how much oxygen saturation. So, you have to take such a spectra, deconvolute it to find it out, because UV visible spectroscopy is quantitative. So, you can find it out how much oxy, how much deoxyhemoglobin that we have, right. Now, as you know, hemoglobin has this cooperative state, cooperativity in the binding curves, that is a sigmoidal, and this is a hyperbolic curve where there is no cooperativity. So, binding of oxygen alters the environment of the heme, right. It goes into different coordination geometry. So, therefore, altering the electronic state level because of the binding changes the environmental, and this can change. Now, visible spectrum can be used to monitor what is the binding state. So, oxyhemoglobin responsible for bright red color of the oxygenated blood, and that is what essentially you look at how what is the color and how it changes, right. That is what we do. Again, another molecule that we discussed, FMN or FAD, these are important constituents of many enzymes that participate in our electron transport chain or redox reaction. So, you can also find it out the action of your electron transport looking at the UV visible, right, whether it is FADH, it is a protonated or deprotonated, here is a quinone group or semi-quinone group. So, their absorption maxima is changing, this is 570, this is 490, right. So, here is 450. So, many of such oxidation state or change of these molecule can be absorbed monitoring the simple UV visible spectrum. Now, how the effect of acid changes the UV visible spectrum emission property. One example I am giving you for curcumin. You know, just I will pause for a minute to explain the curcumin. Curcumin, all of you know where it is found? In haldi, and we use haldi for various application. It is one of the important like a molecule that has been implicated in many diseases, using like a Parkinson's and neurodegeneration diseases and all those. So, getting this curcumin, the high percentage of curcumin is very important, and that dictates the quality of a haldi. All of haldi will have curcumin, but they of different level, and that is that depends upon what is the soil condition, what is the cultivation state, what is the temperature where it is grown. So, the quality of haldi is determined by, quality of turmeric is determined by what is the percentage of this curcumin is present, right. That is the medicinal value it has. And now, this is also sold nowadays as a food supplement. So, quality assurance of turmeric can be done simple looking at the UV visible spectrum or you can quantitate by HPLC or NMR to get the quality assurance or quality control of the turmeric, right. So, if it is in RO, like if you look at the neutral pH, this is the spectrum. If it is in basic pH, this is the spectrum, the blue one, and if it is in acidic pH, this is the spectrum of that, right. So, the pH changes the emission spectrum of UV visible spectroscopy. So, even the oxidation state of the curcumin, you can find it out whether it is in basic or acidic or neutral pH, and one can essentially out of these you can quantitate it. It is looking at the absorption of this molecule. You can combine with other orthogonal technique to have a more quantitative estimate, but this is the simplest way you can find it out, the state of and curcumin which is present in the turmeric, right. What are other application of UV visible? So, you see UV visible is the first level experimental tool that one can find it out whether the drug is binding to a protein or not. So, this is a simple drug, say like we are saying ibuprofen, that we all take as analgesic in headache or something. So, when we consume this, when this molecule releases from the tablet, it goes into blood stream and bind to a protein called HSA, human serum albumin. Where it binds, how it binds, one can find it out using UV visible spectroscopy. So, here is, so you are taking the HSA or at different temperature. This is the HSA spectrum at pH 6.5, this is the HSA spectrum at pH 7.4, and then we are doing the same thing, but we added the ibuprofen, and you can see how the spectra of HSA is changing upon ibuprofen binding at both the pH. So, in this plot you can even plot it, the concentration versus the wavelength change or the absorption change, to find it out the binding constant. Now, one can do the comparative spectroscopy analysis of ibuprofen binding with human serum albumin. This is our HSA, and people found that these are the some sites where it could bind, and with a temperature what is changing. So, pH what is changing, or you can see here the change in the spectra that is evident. So, one can quantitatively determine the binding of HSA with ibuprofen, temperature dependent as well as pH dependent, and one can find it out. That is the two applications that I showed you of UV visible spectroscopy in detection as well as the drug discovery or drug design. Now, another important concept that we are discussing, the difference spectroscopy, right, difference spectroscopy. When a nucleotide binds to an enzyme, what happens? The UV visible spectrum changed, and these changes are not very big, they are small. Now, how much change is happening, can we find it out, and that the approach is called difference spectroscopy. So, what we do with different spectroscopy, in one case you have the reference, in another case you have a reference plus say nucleotide, and you record this spectrum with two beams, two cuvettes, one just say enzyme, one enzyme plus nucleotide. Record the spectrum from one beam, one cuvette contains nucleotide, another contains protein, and then one of the cuvette contains mixture of nucleotide and protein. In another buffer you record the spectrum and find it out like this. Absorption is EL, enzyme plus ligand, the concentration individual of these, right, one can find it out, and you solve the equation at different, say A2 is just enzyme and ligand, A1 is the mixture, you take it delta A, you find it out, and by measuring the difference absorption directly measures the concentration of enzyme nucleotide complex. So, essentially what we are doing, we are monitoring the reaction here. So, we in one cuvette we had just enzyme and just ligand, and when we mix it, there is some free enzyme, some free ligand, and some complex has formed. So, we want to know how much that complex has formed by doing this. So, here is the mixture one, this is the pure one, and we subtract the like a pure one from the mixture one to find it out how much the complex is formed when this reaction is happening, right. So, this kind of experiment has been done. The difference absorption spectrum of ribonuclease with cyclic AMP as a pH function. One can see here is the different pH giving different spectrum of the molar extension coefficient versus lambda is plotted, and by looking at these we can find it out how the reaction is changing with different pH and how much it is happening, right. To sum up what we discussed till now, we discussed the multiple component analysis. This essentially is used to determine whether the signal is coming from 1 or 2. If it is from 2, how much contribution is coming from each of these. Can we find it out by principle of additivity, because that says that both these contributions are just additive. And then if the spectra is complicated, one can use the statistical approach for analyzing this multiple component. We also looked at depending upon binding whether red shift happening or blue shift happening. I also gave you some of the example where we can use UV visible spectroscopy for drug discovery, for diagnostic, also for complex formation. So, after giving enough example and concept of UV visible spectroscopy, we will switch the gear from the next class, and we will look at the other topic that is important for biomolecular spectroscopy. So, this I am logging off for today. Thank you very much.